Right, well, thanks for joining me once again, where people continue to be wrong on interwebs, don't they? They continue to be recidivist defenders, like this clown here, Jay Feldman, who's a very young man with a vastly overinflated idea of his own competence in biochemistry for some reason. Let's hear what Jay has to say about, well, his video is entitled, Why You Want to Burn Carbs, Not Fat, the Research. Well, no, Jay, you don't want to be burning carbs instead of fat. That's the last thing you want to be doing, actually. Anyway, let's see what your opinion is and whether it's any good. Spoiler, no. By the way, I haven't watched this through. I don't know what he's going to say exactly, although I do, because it's the same thing every time from these clowns. So this is off the cuff entirely. Right, off you go, Jay.
So when we're on a low-carb diet, when we shift away from utilizing carbs as a fuel, we see a much greater reliance on fat as a
what a shock.
Fuel for the vast majority of our bodies. But there are certain areas that normally require glucose to function, but they...
Yes, that's why gluconeogenesis exists. That's why it's a thing that's all taken care of. After 3,800 million years of evolution, the human species has this down, Jay. What's your problem? Other than a very severe ego problem, Dunning-Kruger, and a vastly overinflated view of your own competence.
They can't actually use fat efficiently, and so they will use ketones. We'll start to produce ketones in these instances for those different areas. So in general, our bodies are mostly running on fat if we're not eating any carbs, and there's certain portions that are utilizing ketones, and there are certain portions that are still utilizing glucose that actually can't use fat or ketones. And,
right. Okay. So?
And the biggest concern here is that fat as a fuel is far less efficient than carbohydrate as a fuel. Than
According to whom, on the basis of what? Define efficiency on what scale, by what metric, Jay? I presume you're going to attempt to do that. I hope so. Otherwise, you'll just be giving us your opinion again, Jay. And once again, let me give you my take on opinions. They are just like anal orifices. Almost everybody has one, that is, and it's almost certainly full of shit at any given time, Jay.
Glucose as a fuel. And what we basically see, we'll dig into the details, but what we see on a broad sense is that fat oxidation or fat burning leads to slower, less efficient ATP production.
Slower? Why is that a problem? Do cells ever become short on ATP? Spoiler, no, they don't. It's a self-protecting thing, Jay. Cells will not particularly change their concentration of ATP. It's tightly regulated, it's highly controlled. If there were an influence by which a cell would stop having enough ATP and the ATP concentration would drop, there are built-in self-protecting mechanisms that stop that cell from spending ATP at that time, precisely in order to protect it. So slower might not be a problem. You haven't shown us that it is a problem. And efficiency, you still haven't defined. What do you mean by efficiency, boy?
Slower, less efficient oxidative phosphorylation or mitochondrial respiration.
And so slower mitochondrial respiration is a problem, Jay? That's what you're implying. Why is faster always better? I'm told it's not, necessarily.
Higher rates of reactive oxygen species production and higher oxidative stress as a result.
Yeah, okay, so why is that a problem? If that were, in fact, indeed demonstrably true. Those are signaling molecules, Jay. They give an ability for cells to signal their energetic state so that other cells can work in concert, so that the organism can work in concert to maintain its homeostasis. Furthermore, there are a suite of antioxidant molecules that the body produces itself to take care of any deleterious problems that may arise from having a level of antioxidants or free radicals in the body that is higher than what the body can sustain and deal with. There's still no problem here that you've identified. You're giving us partial bits and bobs of information that standalone are correct bits of information, but the logical leaps of faith you're making from stone to stone to stone here are so vastly wide that the real, very real danger and the reality of the situation is that you're going to end up falling in the water sooner or later, son. Because every step you take to the next stone is a leap of faith that's so wide it's laughable. Faceplant. What's next?
Result. And this less efficient energy production.
Again, you haven't defined efficiency, Jay, so you can't tell us it's less efficient. Sorry about that.
Is fine for low energy demands. So if we're talking about the muscles at rest, they don't have high energy needs. It's like a low energy demand state. There's not much activity going on. So in a state like that, fat is fine to be used as a fuel, even though it's less efficient.
And the utilization of fat by muscle cells doesn't particularly, like everyone will argue that as the intensity spectrum goes up, exercise intensity increases from rest to volitional exhaustion, that the relative contribution of fat to energy production drops, which is true. It does. What about the absolute amount of fat? Because even when the RQ or RER is well above 1, we are expected to believe that the contribution of fat when the RER reaches 1 is 0%, and that's not correct. There are confounds to that number, which, people have written and published research papers about, including myself, Jay. Have you ever done that? Published any research articles at all on human physiology whatsoever? No? Okay. I have. And I can tell you that the RER value being above 1 does not indicate that the relative contribution of fat to the oxidation in mitochondria is 0. That's false. Absolutely false. There are several things that artificially raise the RER value and uncouple it from the classic math that they use to determine the contribution relatively, is what I'm saying. So again, leaps of faith. This is Jay Feldman mindlessly repeating things that he's been taught by someone who also doesn't understand, who probably also hasn't published any peer-reviewed articles of their own either, who probably hasn't contributed to knowledge either. Anyway, go on though.
But when we look at a tissue like the brain or the nervous system, there are extremely high energy demands, and the nervous system is very sensitive to damage and oxidative stress.
That's why we have the suite of endogenous antioxidants to prevent that problem from occurring, pretty much, Jay. Okay, what's next?
And so this is a system that can't actually use fat as a fuel. And this is a system that has to rely on glucose or a combination of glucose and ketones. And so that detail is going to become important here.
Is it? I'm glad you're pronouncing the T in important, though. It's very important that people pronounce their words properly.
In terms of why, or when we look at the physiology, it becomes clear as to why fats are not used as a fuel in the brain, but ketones or glucose are. I just wanted.
It'd be very interesting to see what your opinion on that is, Jay. Remember about opinions? We spoke about those a few minutes ago.
At least start by looking at these studies in different.
Oh, the studies, good. I'm glad we're going to look at the studies. Good.
Animal and human models showing what happens with fat metabolism versus carb metabolism. Okay. So I'll just go through both of these to start. The first one is a paper titled Palmitate-Induced Changes in Energy Demand Cause Reallocation of ATP Supply in Rat and Human Skeletal Muscle Cells. And they state probing the bioenergetics of rat and human.
So what you're going to do is give us the title of a paper and give us one sentence out of that paper and claim to be acting as a scientist now, are you, Jay? Because that's not what scientists do. At all.
In myoblasts in real time, we show here that the saturated fatty acid palmitate lowers the rate and coupling efficiency of oxidative phosphorylation under conditions it causes insulin resistance.
Do you see how there's still no definition there of efficiency? There's also no definition of insulin resistance, which is nothing more than a fallacious construct in the way that it's communicated at large, out there in the marketplace. Neither of those things are defined at all by that sentence. I hope you're going to give us more from this paper. Let's see if you do.
What we're seeing here, they looked at both rat and human skeletal muscle.
Yes, we could see that because it says so, that in the title. They looked at them, but what did they look at, Jay? What was done by these authors? What experimental protocol was undertaken? What was the methodology? What was its flaws and foibles? What were the results? Let's get to that.
And found that palmitate was basically reducing the efficiency.
So now you're just repeating what you've already just said, given that efficiency still hasn't been defined. The metric on efficiency still hasn't been proffered. Let's move on to providing the grist rather than just the vox pop sentence out of construct, sentence out of context.
Of ATP production. In this study, this is a study titled...
Oh, you're not going to do that? Now you're going to flash another study up. I hope you're going to go back to these studies and give us more than just the title and one or two sentences completely out of context, with no definitions of operationally what was done, how was it measured, what was the metric, any of that. Goodness.
Inhibition of NADPH oxidase 2 or NOx2 prevents oxidative stress and mitochondrial abnormalities caused by saturated fat and cardiomyocytes.
So what's your problem then? A homeostatic mechanism that prevents the problem that you're purporting to be a problem, or it works against it, attenuates the problem.
Yeah, the heart mitochondria here. And they state the saturated fatty acid palmitate causes a decrease in mitochondrial respiration in cardiomyocytes. Palmitate causes an increase in both total cellular reactive oxygen species and mitochondrial reactive oxygen species.
Yeah, so what increase? What numbers? You see how there's no numbers there? Neither is there anything by way of operational definitions of anything. It's just noise, isn't it, Jay? Just like everything that comes out of your ignorant, childlike mouth. You're a mental midget, son. You're out of your depth completely. You have absolutely no idea how to apply bits and bobs, reductionist pieces of not incorrect information, but you have no idea of how to put those things together to build a valid working model of the objective reality of how a human works, maintains their homeostasis, and what's what. And you're certainly not providing us with anything whatsoever that would lead us to believe that the oxidation of fat is a bad thing. Because it's not. We've been doing it for, millions of years.
And so I should be clear here, we're not just looking at the mitochondria, we're looking at the entire cardiomyocyte, the entire cell, and seeing an increase in ROS in the mitochondria and outside of the mitochondria.
Do you see how you still didn't give us a number? What do you mean by increase, Jay? And what are the ramifications of that, in your opinion?
From utilizing the saturated acid palmitate as a fuel. And they go on to state, cardiomyocytes treated with palmitate have increased total and mitochondrial reactive oxygen species.
By how much? Once again, see the lack of a number there? Complete lack of any number whatsoever? Nor any statement about the ramifications of that, in their opinion?
So again, we're seeing this play out in the mitochondria and in the cells when they're respiring on fat as a fuel.
Jay, how much time have you spent in a lab actually looking at mitochondria doing anything? What is your exact exposure to actual science as a working scientist? We see this, says Jay. No, Jay. What you do is read papers that you're completely incompetent to read, jump to conclusions that you're completely incompetent to make, that have absolutely no basis, jumping from stone to stone to stone, with gaps so vastly wide that, as I said, you're going to end up falling flat on your face sooner or later, and you've already done it many, many times. This is par for the course for this boy.
And we see this in some other animals as well.
Well, let's look at other animals, because absolutely it's completely valid to look at other animals and make statements about humans, isn't it?
So this first study is looking at pigeons.
Oh, great. Well, pigeons are very similar animals. Good. Imbecile.
We won't go into detail here. I'm just going to comment on it real.
Well, why bring the thing up at all if you're not going to go into detail? That's the whole point. The details. Boy.
Briefly, you mentioned the title, which is...
Oh, just briefly mention the title. I could briefly mention the title of all sorts of things, couldn't I? Does that make my argument any stronger on any topic? Child? Does it? No.
The mitochondrial generation of hydrogen peroxide, general properties, and effect of hyperbaric oxygen. And in this study, they looked at pigeon...
Right, so they're looking at a completely different animal, not humans, in a completely unnatural situation, hyperbaric oxygen, and telling us something about mitochondria and how they function in the real world in humans, are they?
heart mitochondria, and they provided them palmitate in the form of palmitoyl carnitine. So it's palmitate bound with carnitine so that it could be used in the mitochondria. And they found that it increased reactive oxygen species. So this is an
increased by how much, Jay? And what are the ramifications of that still? See, there's still no numbers there that you've provided us with whatsoever. You're just assuming that the production of reactive oxygen species is necessarily contraindicated and a bad thing. Is it though? No. It's a signal. It occurs for a reason. It's dealt with, it's kept under control, it's kept within homeostatic limits by the existing homeostatic mechanism. That's why it's there. It is a signal. It's supposed to happen. When you understand the Randall cycle, because you don't yet, Jay, you think you do, but you don't, you'll understand that. In the meantime, let's hear some more of your opinions, just because they're hilarious.
And pigeons were seeing that. And in this study, we're looking at cows, and this is
cows indeed are very similar to humans, just like pigeons are, aren't they, Jay? Of course they are. What am I talking about? What a buffoon I must be.
A study titled Nuclear Factor.
We can read what the title is, Jay. Are you going to tell us anything worthwhile out of the thing? I doubt it.
Erythroid-2 related factor 2, that's NRF2, protects bovine mammary epithelial cells against free fatty acid-induced mitochondrial dysfunction in vitro.
Right, so cows also have a homeostatic mechanism as well that protects them from the damage as well, do they? Goodness gracious, what a shock. I'm so surprised. What's next?
And this study states...
Remember, this video is entitled Why You Want to Oxidize Carbohydrate and Not Fat. So far, we've had the title of several papers which tell us nothing about anything, several sentences out of those papers which tell us absolutely nothing because they contained absolutely no numbers, nor any statement of the ramification of any numbers that weren't given anyway. And now we're talking about pigeons and cows. Why do I get cross? I wonder. Why do I get terse with these imbeciles? I wonder. Hope you're enjoying the show. Stay tuned. There's more of it to come.
Results indicated that exogenous free fatty acid challenge induced linear and quadratic increases in concentrations of mitochondrial ROS. So essentially, as they increased fatty acid availability and increased fat metabolism, there was a linear and quadratic increase in ROS production in these mitochondria.
Yes. So that's the design. It's supposed to happen.
Again, we're looking at the mitochondria in cows in this case.
That's right, in cows. Same thing happens in humans too, by the way. And so what? It's supposed to. It's a signal still.
And they go on to state high concentrations of free fatty acids, similar to circulating free fatty acid concentrations.
What is meant by high concentrations, Jay? High according to whom, on the basis of what?
Concentrations in dairy cows with fatty liver during the transition period induced mitochondrial dysfunction in bovine hepatocytes in vitro. So they're talking about other studies.
So when you vastly, hugely, unnaturally increase the concentration of free fatty acids being presented to the mitochondria of cows in a test tube, the mitochondria tend to falter somewhat. Well, so what? That's no surprise. Let's get back to what happens with cows trotting around in the field eating grass, shall we?
Study showing that you saw the same effect in liver cells from the cows, as well as in this study when they were looking at the mammary epithelial cells.
Wow.
So,
all right, so take a car that runs on 91 RON octane gasoline, punch in a bunch of 98, and run the thing for a bit. See what happens. Back in the real world, though, the thing is designed for 91, and it runs fine on that. We done? What's next?
we're seeing these same mechanisms.
The mechanisms, boys and girls. Let's hear about the mechanisms, Jay.
Throughout species, in different tissues as well, that all are used to utilizing fats.
So you feed somebody, be that somebody person or a somebody animal, a diet completely inappropriate for them, and there might be problems. Goodness, who'd have thunk that?
When they utilize fats as a fuel and you increase that fat utilization, you see increased ROS and less efficient ATP production.
What do you mean by efficient ATP production? Still, boy. Yes, you see increased ROS. That's supposed to happen. Still, that's the design. Still, and you haven't defined efficiency. Still, neither have you given us any reason to believe that reduced efficiency is a problem within the homeostatic range. Still, what's next?
And then we have a number of studies. I mean, I'm sure.
Oh, a number of studies. Cite them. Boy. No? Okay.
A fraction of all these studies here. I'll be citing more in the show notes, but these are just some of the more notable ones. But in this case, we can look at studies that are looking at exactly how these ROS are produced, how the reactive oxygen species are getting produced in this scenario.
Yep. You could if you like, but would that tell you that those ROS, by very virtue of being produced, are necessarily a problem in a free living human being living the way it ought to, according to the natural design, Jay? No. Still.
How efficient the ATP production is and how
again, define efficiency. Still.
Exactly that's happening. They're able to break it all down by, blocking certain complexes or zooming in at, basically what's happening and what products are.
Right, so a bunch of reductionist and probably not incorrect information that he's about to give about some of the mechanisms by which the ROS do indeed build up when beta oxidation prevails in human mitochondria. To which I'm going to respond, so fucking what? Let's do that.
Are being produced. And so we can see this all very clearly. It's all very well documented. And we'll take a look first.
Yeah, but Jay, you're not competent at all to decide what is well documented, what isn't, what is science, what isn't good science. You've never produced any science yourself. You've never been involved in peer-reviewed publications, as far as I'm aware, or in any topic regarding to biochemistry. You're a very, very young man with a vastly overinflated view of your own competence. You have Dunning-Kruger dripping from every pore, or is that greasy, fatty substance? It looks more like that. Why are you so shiny, son? Anyway, let's hear more about this ROS, though.
The study titled Topology of Superoxide Production from Different Sites in the Mitochondrial Electron Transport.
Yes, we can read what the title says, Jay. Why don't you tell us more about the paper in an actually competent, critical fashion, dealing with the methodologies of the study, its flaws, its foibles, its faults, its strengths, the outcome statistics, and the ramification of those. No? Crazy talk from me. Okay.
Chain. And in this case, they looked at skeletal muscle,
Yep.
They looked at heart, they looked at liver cells, and the mitochondria there, and they basically provided fatty acids and saw that there was far more reactive oxygen species being.
Yeah, we still expect that, Jay. It's still exactly in line with what the design is, what's supposed to happen, and what we expect. What's your problem? Other than Dunning-Kruger, a vastly overinflated belief about your own competence in biochemistry, and a lack of, well, really, any kind of wisdom or experience in the field whatsoever.
Being produced, and in this case, this is in a rodent model. And
oh well, great. Are we done? Because the title of this little film by you, Jay, is Why You Want to Oxidize Carbohydrates and Not Fats. I'm not a rodent or a pigeon or a cow. Okay?
And so we'll just look at a couple of quotes here where they just.
So to boil the whole thing down to how mitochondria behave is reductionism of the worst kind. Because I think you'll find that pigeons, cows, and human beings are all very different animals, aren't they, Jay? There might be similar mechanisms going on in the selection of fuels in terms of the ROS production commensurate with beta oxidation as compared to the cleavage of glucose or AF or acetyl coenzyme A. But what you haven't done, and what I suspect you're not going to do because you're running out of time in this video, is to give us any idea at all as to why we should see the beta oxidation of fatty acids is in any way deleterious. At all. Let's go.
describe in detail how this is all happening.
Yes, well, okay, good. Let's hear the details because it's interesting, but that doesn't provide for the title of your video. It doesn't inform on the title of your video because actually there is no argument extant which can support the title of your video, which is, once again, why me, and you, and everybody else who's human, I presume, because this is a video for human people, why they want to oxidize carbohydrates and not fats, which is not true, Jay. It's false. They don't want to do any such thing. Off you go, though.
Already went through this in terms of the mechanisms, but...
Oh, yes, the mechanisms. Did you, though? Who's clearer now on the mechanisms than they were before we started Jay's little video? I'm not. I was already pretty clear, mind, but there you go.
Touched on it before, but this is the research that's supporting it, that's saying this is actually what's happening. And
right, well, we know it's happening, though, Jay. That's no surprise. We absolutely know that when beta oxidation predominates in any given mitochondria, that mitochondria will produce more reactive oxygen species. So?
These scenarios. And so they go on to state here, mitochondria from skeletal muscle or heart generated significant amounts of superoxide, that's a reactive oxygen species, from complex 1 when respiring on palmitoyl carnitine. So that's basically the fatty acid palmitate bound with carnitine. They go on to state the greater hydrogen peroxide production with palmitoyl carnitine than with complex 1 substrates could be because complex 1 is more reduced with palmitoyl carnitine due to reversed electron transport and competition with the ETF.
Yeah, you said could be there. That leaves maybe not 2 in terms of the actual mechanism completely on the table, and it makes no statement about which one's more likely, does it? That's the great thing about science when you write could be this, maybe that. Establishes nothing.
Ubiquinone oxidoreductase for oxidized Q. So I'll just pause there, but they're describing exactly what we were seeing earlier. With the fatty acid metabolism, you have far greater drop-off of electrons from FADH2 relative to NADH compared to glucose, where you have far more NADH electrons.
Yes, we understand the mechanisms, those of us that do. Yes, that's correct. That is how it works. Extremely parsimoniously, by the way that you're describing it, yes. What you're not doing, Jay, is giving us any ramifications of that or any reason to believe that that's a bad thing, because it still isn't.
Drop off relative to FADH2. And in this scenario, they're describing exactly what we were describing earlier, which is that they saw increased reactive oxygen species.
Yes, we still know it's still a thing and it's still not a problem. What's next?
Due to potential reverse electron transport.
Potential. See, there's the word again. Maybe, possibly, potentially, could be. Maybe not too. Has there been any actual reverse electron transport measured, Jay, by anybody? If so, how so? And when so? Give us that paper.
And also the competition due to basically the fact that all of the coenzyme Q gets reduced and there's not enough oxidized Q left or ubiquinone left. They go on to state greater steady state reduction of complex 1 would lead to greater matrix ROS production with palmitoyl carnitine as a substrate. Additionally,
which we still know about, and it still isn't a surprise.
Could be that the ETF, ubiquinone oxidoreductase, and ETF can also produce superoxide on the matrix side of the membrane when palmitoyl carnitine is added. So we saw this in that earlier diagram that there's actually ROS being produced at those enzymes. And they go,
no, we didn't. We didn't see anything of the sort. You said that, and I'm not going to disagree with you on it, but I'm also going to challenge you to provide some empirical evidence that it does occur, measured somehow. I'm quite happy for that to be the mechanism, if it is, to which the response is still going to be, so fucking what?
You go on to state fatty acid beta oxidation does lead to release of reactive oxygen species.
Yeah, we still know, and it still isn't a problem. Still.
Primarily from complex 1 on the matrix side of the inner membrane, which can lead to hydrogen peroxide spilling out into the cytosol. So that's what they found to actually be the case.
Can, and perhaps not too. Maybe there's homeostatic mechanisms that prevent the spillover of hydrogen peroxide. Like, I don't know, H2O2 dismutase. Oh, yeah. Good. What's next?
We'll go on to this next study, which is titled...
That's why H2O2 dismutase evolved, by the way, to deal with that problem, Jay. That's what it's there for. Okay? What's next?
Electron transport chain dependent and independent mechanisms of mitochondrial hydrogen peroxide emission during long-chain fatty acid oxidation.
Good. Go.
So again, similar study, in this case looking at a different rodent model in mice.
Mises and mices again? I hate mices to pieces, frankly. Got any human studies, Jay, at all to look at? Even if these mechanisms are identical in humans, even if the response remains exactly the same, doesn't it, boys and girls? Let's say it all together. So fucking what?
Seeing those same effects and identifying where these ROS are coming from when the cells and mitochondria are utilizing fat as a fuel.
How about instead of looking where they're coming from, how about looking at whether or not they pose any problem ongoingly to the human system? Because the answer is no. It doesn't. Homeostasis is still a thing. Still works very well.
Fuel.
The increase in FADH2 versus NAD under beta oxidative influence is expected, is known about, is a signal that the body uses to communicate its energetic status. It is a coordinating influence over the cells of the body. It is fine. It is dealt with by a whole bunch of endogenous antioxidant molecules that the body produces itself, precisely to protect itself from any damage that might occur in that case. It's not a distress at all. We have evolved for millions of years, oxidizing fat primarily as our main source of energetic substrate. The mitochondria have had a very, very, very long time to get their shit together in this respect. They've done so. What is your problem other than an ego problem and Dunning-Kruger? Boy.
They state, using skeletal muscle mitochondria, our findings indicate that even a low supply of long-chain fatty acids is associated with ROS formation in excess of that generated by NADH-linked substrates.
We know still. Yes.
So the NADH-linked substrates being, glucose and pyruvate, whereas we have more of an FADH2-linked substrate here with the fats.
Yes, correct. Still.
And so this was associated with greater ROS formation. They go on.
Yes, still. We still know it still isn't a problem.
To state,
you're running out of time, Jay, to tell us why this might be a problem. Your video's nearly finished.
Moreover, ROS production was evident across the physiological range of membrane potential and was relatively insensitive to membrane potential changes. Determinations of topology and membrane potential, as well as use of inhibitors, reveal.
Isn't that interesting? What does that tell you? If you're thinking, which I know you're not capable of, all you're doing is regurgitating reductionist nonsense. What you're saying is factually correct. What you're not doing is linking it together in a logical way that shows us you understand the integrated function, the homeostatic function, the design of the thing at all. You're a silly little boy. Let me help you out with this. If this is relatively insensitive to the membrane potential changes across the physiological range, it tells us that it occurs by design as a signal. No? Okay. What's next?
Revealed complex 3 and the electron transfer flavoprotein ETF and ETF oxidoreductase as likely.
Gee, I wish I knew as many big words as this boy. I do. But you see how actually he finds it necessary to obfuscate his responsibility if he's going to make public statements about how things work. Instead of telling us how they work, what he's going to do is list off his vast knowledge of big words without any comprehension of the meaning, interaction, and workings of any of it. This is clearly a boy who knows how to memorize things well. Not as well as I do. I'm a bit of a world expert in that, in my own lunchtime. But nonetheless, this boy is vacuous in terms of his comprehension of what it is he's actually talking about. It's nuts.
Sites of ROS production.
Good.
So they were able to identify, we know there's ROS, where is it coming from? And by blocking, they mentioned using different inhibitors and, using different techniques in the research, so they're able to identify this is actually where it's coming from. And that's how we end up with these pathways, is we actually see that they're happening in these sorts of studies. And then
we know, Jay. So what? Still.
We identify, okay, we know that when we're oxidizing fats, we're getting ROS from these places.
Right. Which is the design. It's a signal. It's supposed to happen. It's not a problem. It's dealt with. What's your point?
Here's a couple of other studies.
Oh, God.
Showing similar effects. So
these ones human studies?
This is a study titled Oxidation of Fatty Acids is the Source of Increased Mitochondrial ROS Production in Kidney, Cortical Tubules, and Early Diabetes.
Yeah, we know.
So in this case, they induced type 1 diabetes in rats.
Okay. We done?
And they were looking at mitochondria in the kidneys. And at first they state, owing to kidney cellular complexity, the current study was performed specifically on mitochondria isolated from cortical tubular structures.
Oh, right. So isolated mitochondria. Do you think isolating mitochondria from the cell environment would cause it to behave differently? If you don't, I think you better go back to school. It does. This is an invalid model even to rats, let alone to human beings who have different nutritional requirements, whose mitochondria have evolved specifically along very different nutritional lines over many, many millions of years. You pathetic, ridiculous, arrogant little boy. I am sick to the back teeth of these ridiculous little cunts coming online, making out like they're some kind of expert because they can say words bigger than wheelbarrow. Not remotely anywhere near good enough. Jay Feldman, shut the fuck up. Shut down your ridiculous fucking channel. Get your ridiculous fucking ass off the internet. Go to school and actually fucking learn something, rather than spending your whole life memorizing bits and bobs of the metabolic machinery in complete fucking isolation to how they work together. Jesus H.
The highly oxidative nephron segments that depend on the ATP generation from fatty acid oxidation, free of medullary structures which are highly glycolytic. So they're just identifying they were specifically using mitochondria that normally use fats as a fuel. They were making sure not to use the mitochondria that normally are just using glycolysis as a fuel because that wouldn't be a good representation of what goes on with fat.
And so what they did was remove mitochondria from the cells in which they live, which makes them not a good representation of fucking anything whatsoever, because that's not how mitochondria exist, is it? They interact with the cells in which they live. They react to those cells in the same way that those cells react to them. They modify each other's behavior. We're done. What's next?
Fatty acid metabolism in mitochondria.
Bring the next study up. Let's go.
And what they found looking at those mitochondria was that...
Who cares? Next time I'm talking to a rat about their mitochondrial health, I might be interested in this study. I probably still won't be because it's still on isolated mitochondria. Neither will the rat be interested because they don't even fucking know what a mitochondria is.
Mitochondrial fatty acid oxidation is the source of the increased net ROS production.
We still know that. And we still don't care, those of us who understand. Which doesn't include you, Jay.
And the site of electron leakage is located proximal to coenzyme Q at the electron transfer flavo.
How was that located? And is that valid when you have removed that mitochondria from the cell in which it lives? No.
Protein that shuttles electrons from acetyl-CoA dehydrogenases to coenzyme Q. Oxidation of fatty acid substrates was reported to be a source of ROS in the normal heart and skeletal muscle mitochondria. Increased oxidation of fatty acids in aorta.
So it's basically, this video says you oxidize fat and it's likely that you'll produce more ROS. Well, we know that, Jay. Still. Completely pointless video, and it does not establish your point. The assumption that you're making is that reactive oxygen species are necessarily contraindicated, negative, and a problem. They're not. Your knowledge is vacuous in the field of physiology. Your understanding of the holistic functioning of the human body is vacuous. Vacuous. You are an imbecile. You are an arrogant Dunning-Kruger sufferer who needs to shut the fuck up. Simple. What's next?
Aortic endothelial cells was reported to increase superoxide production from sites located within the electron transport. So we're just talking about other studies here, looking at this in other cells, other tissues, and other sources of ROS production. They go on to state the equal decrease of ROS with rotenone indicates that an additional site rather than the reverse electron transport to complex 1 is responsible for the increased ROS generation by diabetic tubule mitochondria oxidizing fat substrates. So basically by using certain inhibitors here, they were identifying that it's not just reverse electron transport driving the ROS production, although that can be a factor as shown in other studies, but they were finding that there are other places where we're seeing this ROS generated. They go on to state these data also indicate that a site other than complex 3 significantly contributes to ROS generation when fatty
Oh, fucking pot. Still.
acids are used as substrates in diabetes. So there is ROS generation at complex 3, but they're saying there's another site as well.
We heard you the first time. Still.
And they go on to state the study identifies fatty acids as the source of reducing equivalents.
We still know. We still are not interested in this because it still does not present any issue.
Responsible for increased ROS production by kidney tubule mitochondria in diabetes and shows that ETF, the electron transfer flavoprotein, is the major site of electron leakage. So in the study, they were showing that that's where most of the ROS are coming from. But again, this is just another study demonstrating that we can see very clearly that when we're oxidizing fats, this is actually occurring. We are seeing an increase.
Nobody's arguing that, Jay. Still, that does occur, yes. It's supposed to still. It's a signal still of utility still. It occurs within a magnitude that's not problematic for the longevity of the cell. Still, the homeostatic mechanism takes care of it. Still, what is your point? You still have not provided us with any reason whatsoever why we would want to oxidize carbohydrate instead of fat. Still.
Increase in ROS directly correlational with the increase in fat metabolism. And we know exactly how this is happening in the mitochondria. We can see exactly where it's coming.
No, not exactly. You've given us a bunch of theories, a bunch of animal models, and isolated mitochondrial models of other animals. You've given us an outline of the theory as to where these reactive oxygen species are generated. That's not really the point. It doesn't matter how accurate those things are or not. The point is it still is not a problem, Jay Feldman. Imbecile.
From. And this last study is looking at uncoupling, which I touched on earlier. We'll touch
right. Uncoupling protein. This will be interesting.
On it a bit more here. And this study is titled Induction of Endogenous Uncoupling.
We could read the title ourselves still, couldn't we?
Coupling protein 3 suppresses mitochondrial oxidant emission during fatty acid-supported respiration.
It's almost like that sounds like a homeostatic mechanism that works against this issue. Goodness.
And they state, uncoupling protein 3 expression increases dramatically in skeletal muscle under metabolic states associated with elevated lipid metabolism.
Goodness gracious, homeostasis. Who'd have thunk it?
Yet the function of UCP3 in a physiological context remains controversial.
So what does that mean? Controversial. It means that there are different opinions about it. We've covered opinions already, haven't we, Jay?
Elevated uncoupling activity was evident in the EXR fibers only upon the addition of palmitate, which is
again backing up the idea that this is probably one of those very effective homeostatic mechanisms protecting the cell from such problems. Yeah.
is a known activator of UCP3 or under substrate conditions eliciting substantial rates of hydrogen peroxide production, such as respiration supported by succinate or palmitoyl L-carnitine malate, but not with pyruvate malate.
Right.
Indicative of UCP3 activation by endogenous ROS. In this study, they were actually not just looking in vitro at isolated mitochondria, they were looking at rat and mouse muscle fibers.
Right, so next time I'm talking to rats and mices and rats, I'll be sure to point this study out to them to help them to understand, once I've taught them how to speak English, that in fact the homeostatic mechanisms in mices and rats, probably very similar to those found in humans. The uncoupling protein there, number 3, looks like it deals with the H2O2 problem. Well, fuck me. You fucking imbecile. You absolutely ridiculous fucking child. Go and do your fucking homework, for Christ's sake.
This is what's called an in situ study. So it's more relevant to what happens in vivo as opposed to just looking at mitochondria individually. What they basically found was that with elevated fat metabolism, and specifically with using palmitate
Oh my God, really?
as a source of, as the fat metabolism source, they found increased ROS production, and they found that this increased uncoupling activity, which, as I touched on earlier, is something that helps the cells deal with increased oxidative stress.
Right. So tell us once again why it is that we should avoid the oxidation of fat then, Jay. Given that you've just outlined the mechanism, or one of the mechanisms, by which homeostasis is maintained, and this is not a fucking problem. So we wait for that.
By basically stopping ROS production because it dissipates the gradient in the mitochondria, but as a result, you also get less ATP production. And so
Cells have a protective mechanism that means their ATP concentration doesn't drop very much at all. Yes, they do. There's another mechanism that you're clearly not aware of. You really are an absolute cocksplat, aren't you, boy? You're a complete waste of perfectly good oxygen. You have no business whatever professing to know anything about holistic integrated functioning of the human metabolism whatsoever. This is absolutely fucking ridiculous beyond belief. Thank God you nearly finished.
you saw this playing out here in this study. So on the whole, we have a huge body of research demonstrating that these
A huge body of research is a value judgment statement. There's nothing scientific about it. It's your opinion, Jay. What do you think it's showing?
pathways I was talking about, the effects of fatty acid metabolism, do directly happen. We know that these are actually what happens when we're metabolizing fats.
Yeah, and nobody's arguing with that. We know that still. But you haven't given us any reason whatsoever to avoid that because it's normal, indicated, and of utility. The homeostatic mechanism still takes care of it. My word, the arrogance of these children. I blame the parents, frankly. I blame Mr. and Mrs. Feldman. You need to do better, people, at raising your children to understand their limitations.
We're not talking about some kind of random obscure pathway that's leading to some.
You gave us some pretty good theories on it, which there's no point in arguing with because it doesn't fucking matter where it comes from. God's sake.
An increase in some metabolite that could be harmful. We're talking about the
Could be, but it's not because it's dealt with by the homeostatic mechanism still. Oh, fuck me sideways.
basic function, the basic way that cells utilize fats as a fuel, including the cells that are basically designed to utilize fats as a fuel.
Which is all of them pretty much. There are very few cells that don't in terms of the different cell types. There are large numbers of cells that don't use fat, like brain cells. Sure. Testes. Well, some have more cells than others in that regard. That's for another day.
In those cases, we still see this happening. We see it not only in rodents, not only in humans, not only in pigeons, we see it in cows. I mean, this is something that's conserved throughout species and is basically.
You've only named about 4 species there, Jay. Did you make a statement about all species on the basis of looking at 4 of them that indeed all show the same signaling mechanism, which is still of utility and still indicated and still not problematic in any way, shape, or form to those animals? No? Okay.
Built into the nature of fat metabolism. If you enjoyed that clip,
I did not. No, not a bit. Not one bit at all, Jay.
Definitely want to download the free Energy Balance Food Guide.
Well, on the basis of your complete lack of understanding of anything worthwhile knowing, Jay, why would anyone take you remotely seriously and take any advice from you on any topic? Somebody explain to me why somebody might want to do that other than because they're even stupider than Jay is.
The Energy Balance Food Guide will help you determine exactly what to eat.
No, it won't. It'll give you Jay's opinion, which is ridiculous, vacuous, facile, ignorant, arrogant, and dripping with Dunning-Kruger.
To optimally support your metabolism.
Nonsense. Jay doesn't understand metabolism. He understands various different little bits of it in isolation to the holistic functioning of the whole thing.
And help you lose weight, improve
Nonsense.
your digestion.
Nonsense.
Get amazing sleep.
Nonsense.
Boost your energy.
Nonsense.
And so much more.
No, nonsense.
The food guide makes it extremely easy to get started with a
that's, again, an opinion. Extremely easy as an opinion.
Bioenergetic approach to optimizing your health.
No, bioenergetics will not optimize your health. That's, for those who don't know what bioenergetics is, that's the Ray Peat school of thought. It's ridiculous. It's farcical. It's nonsense. It's garbage. It's an area of philosophy, theology, really, that was started by the late Ray Peat, which he's lost control of entirely now because he's dead. And there's a bunch of people who think they understood anything Ray Peat had to say, have interpreted it completely falsely, and are now espousing completely false, ridiculous theology as if it's what Ray Peat actually said. It's a bit of an insult to his memory, really. Not that Ray Peat was particularly sharp. He wasn't the sharpest tool in the shed, and I'm not bigging up Ray Peat at all. A lot of what he said was absolutely ridiculous. But what these guys are saying is actually not what Ray Peat said. Okay? Right, I think we're done. Join me next time when someone will be wrong on interwebs, because it's not slowing down, is it? No. See you then.